name is Olga Kubassova. I am CEO of Image Analysis Group. Today, we will speak about musculoskeletal clinical trials. We wanted to make topic as comprehensive as possible, and obviously it's difficult in 30 minutes to tell you in great detail about bone health, obesity, OA, and cartilage repair. So the presentation itself will be more general, just to give you an overview of the modalities, opportunities in those indications. We will then follow with a live demo of DYNAMIKA, which is IAG's platform for imaging data management and review. So Shreya here on the call will be demonstrating DYNAMIKA. That should give you a very practical insight into how we work within a clinical trial, how platform really supports all of those image modalities from whatever angle you're looking at your data within a trial. And then, of course, Q&A. So I have a chat open just in front of me. So if you have a burning question, please don't hesitate to drop it. And we will come to those questions at the end. Excellent. So we'll move to the next slide. And it's my pleasure to introduce you to today's agenda. So imaging as a strategic endpoint in musculoskeletal trial, we will cover DXA, MRI, X-ray. We will then look at some of the indication-led modality strategies, so what modality to choose and how to use them promptly. We will then look at the practical workflows and quality control within the trial and look at the real opportunities for the CRO to de-risk some of these clinical trial endpoints and workflows. We will finish the call with a case discussion and live demonstration of DYNAMIKA. So on DYNAMIKA, primarily, you will have DXA and X-ray analysis. So we're not going to go into very complicated AI workflows, but the DXA and X-ray are principal modalities in some of the current trials. So we will cover those. And next slide, please. And it's my pleasure to introduce the speakers today. So myself, I will start the presentation. I'm Chief Executive Officer of Image Analysis Group. I'm also the founder. I started the company 20 years ago, and that's where we're still going in the imaging clinical trials. With me today, Michael Clark, Chief Operating Officer of IAG. Michael brings with him 20 years of experience in imaging. Romiesa Hagoug, Head of Project Management, and Shreya Nair, Product Manager at IAG. I will introduce you to IAG. Maybe some of you are new to us, and it's really good to refresh, maybe update on capabilities to those who know us. So as I said, we are 20 years old, nearly 20 years old. We run over 700 trials. We maintain a very extensive network of imaging centers who we work with. From every trial, we have a number of centers that we collaborate. We also have an extensive pool of radiology readers, experts, and KOLs, which work with us on trials. We specialize across multiple indications with key therapeutic areas that we cover being musculoskeletal and rheumatic, oncology, and obesity. Now box by box, so it makes it easier. With IAG, you will find quite advanced imaging infrastructure through our platform DYNAMIKA. So DYNAMIKA is a cloud-native platform. It's SOC 2 Type 2 compliant for security. It's compliant to ISO 13485 and 21 CFR Part 11 standards. DYNAMIKA maintains role-specific workflows. So as the trial starts, you will find yourself with a number of sites, number of readers, project managers, perhaps a CRO, maybe third-party vendors or partners. Each of those teams, to us, is a specific player on a trial. So each team receives a separate workflow. So the sites, they will be able to upload images. Images gets anonymized as the data comes to DYNAMIKA. Data gets QC'd automatically as it just goes from the site to DYNAMIKA. Once the data hits the cloud, a new role becomes active. So a new role is an imaging specialist. This is a team which are doing quality control of the data. The next role would be the reader. As soon as the data is quality controlled, data is read. So each role works simultaneously at all times across multiple trials. So we would run all our trials within the same umbrella, within the same setup, giving consistency and quality, consistent quality during the imaging trial. DYNAMIKA allows us to open API to third parties. It means that if there is an interesting read developed by a new partner, we can bring that natively to DYNAMIKA. We already maintain over 200 read criteria across indications, across modalities. And we put here some industry KPIs that always, of interest to sponsors. So we can put configuration for collect and hold within 48 hours. DYNAMIKA functions across multiple continents, including China. We have 12-hour QC targets. We have about 60% of queries resolution as the data gets uploaded from the site. We have, of course, more KPIs, and happy to talk to you about them after this webinar. So Image Analysis provides end-to-end project management. Our teams are located in the US, Europe, UK, and India, with additional teams in China and Australia. We provide end-to-end support from protocol design, protocol strategy, selection of endpoints, full documentation, site feasibility. We spend a lot of time with the sites, making sure that they can understand the imaging protocol, they can train and support that protocol for the duration of the study. We support central read, either with expert readers, with AI, or a hybrid read. We support data management, QC, and all ADC connectivity to the sponsors. In addition to radiology, we also support cardiac safety and cardiology reads. You will work directly with our experts. So for every study, we would have expert readers, expert KOLs. We maintain adjudication of the read and overview of the quality of the read. So very often that involves an expert. So we work with great reader groups as well as individual readers. We support all regulatory submissions, meetings, letters, and in any geography. Again, any modality with added support for more sophisticated modalities like dosimetry in some of the radiopharmaceutical trials, or handling contrast, or handling the isotopes in radiopharmaceutical trials. Lastly, we have a hands-on AI experience. We have publicly announced partnerships with 5 top pharma. Those are available on our website, and you can come to have a look what AI can do in either real-world studies or in the pivotal studies. I'll pass to Michael to carry on and take us through some of the imaging slides.
Thank you, Olga. So why are we here? Why does imaging matter in musculoskeletal trials? So there's a wide range of things you can get from doing imaging in your trials. And it's not just eligibility, it's not just safety, but you can get eligibility, safety, efficacy, and regulatory endpoints. So in osteoporosis, you might see BMD or T-scores and Genant score in osteoporosis, or you might see KNL graded or JSW, joint space width, in osteoarthritis. In DXA, we're looking for, in safety, treatment-induced bone loss. So you could see in some cases where a subject is on a placebo versus an active asset in osteoporosis, that the patient may be having bone loss. So we want to see that. We want to see that the patient's having bone loss, and we want to intervene as soon as possible. There's other things like sarcopenia, cartilage degeneration, and skeletal-related events like RPOA and osteoarthritis. That's rapidly progressing osteoarthritis. And then we're looking at the objectives and the endpoints. So most or many of the musculoskeletal trials do have imaging as an endpoint or an objective in their trial. And finally, all of these endpoints, or many of them, are primary or secondary endpoints required by the FDA or the EMA. So what are the options that you have to use in musculoskeletal? So there are many options, and the ones that we're going to focus on today are DXA, X-ray, and MRI. But we also have CT scans, PET scans, and ultrasound. Like I said, in DXA, you can get BMD, bone mineral density, body composition, trabecular bone score, or vertebral fracture assessment. And these are in the osteoporosis, obesity, and GLP-1 trials, sarcopenia, and oncology bone safety. In X-ray, you'll see this also in osteoporosis and osteoarthritis. You can do joint space width, structural progression. The limitations with X-ray is that it's limited soft tissue viewing and high positioning sensitivity. And you'll see that in your OA trials and your RA trials. So oftentimes we have to position the patient. We provide positioning devices to help keep the positioning standard across all visits. And then MRI to get a more precise look at what's happening to the cartilage, synovitis, bone marrow in the osteoarthritis trials and the RA trials. We can do CT, we can do PET. We've done PET in rare bone disease, looking at bone turnover. Ultrasound is sometimes used in the RA and the OA trials, sometimes for efficacy or eligibility. So you have a lot of tools in that you can use when doing an imaging trial. Many of them are required by the FDA or the EMA. Some of them you just want to get more information on the drug and how it's and the method of action and see if it's working, or for safety reasons. Choosing modalities is so like I said, you have DXA, X-ray, MRI, CT, PET, and ultrasound. You would often see DXA in the bone health trials, osteoporosis, osteogenesis imperfecta, rare bone disease, and in the obesity trials, more so with the GLP-1 trials that are in the market or in clinical trials at the moment. X-ray, you're going to have in your osteoporosis trials, you're going to have your osteoarthritis trials, rheumatoid psoriatic arthritis trials, and MSK pain in general. So that's often associated with osteoarthritis or rheumatoid arthritis. And then MRI, again, looking at osteoarthritis, cartilage repair, RA, psoriatic arthritis, and ASK and MSK pain. IAG can help you identify what is the best modality and what is the best endpoint for your trial using one of these modalities. Now let's talk a little bit. That was the general overview. Let's talk a little bit about DXA. So with DXA, like I said, it's often seen, it's required in osteoporosis trials. You'll see it in osteogenesis imperfecta, rare bone disease, sometimes osteoarthritis, definitely in oncology trials where there may be an effect on the bone, and that would be in, let's say, a prostate cancer trial. You might see DXA. So in DXA, you can get bone mineral density, BMD, at the lumbar spine, total hip, and femoral neck. And it is a primary measure used to diagnose osteoporosis and monitor treatment effect. You could do T-score and Z-score as an indicator for osteoporosis or osteopenia at eligibility. Z-score is used in for BMD to age-matched norms in younger patients. So it's used in pediatric patients most often, then the T-score is in the older population. Body composition will give you lean mass, fat mass, visceral adipose tissue, android to gynoid ratio. So these are important endpoints for the GLP-1s and the obesity trials that are going on now. So you want to see if you're preserving lean mass, if you're losing fat mass. They also want to see that there's an effect on the VAT, the visceral adipose tissue. So those are important endpoints in the obesity trials. And then there's some more, there's different endpoints that are a bit more, how would I say? Anyways, trabecular bone score. So that's looking at the trabecular bone microarchitecture, advanced, sorry, I got the word. So they're more advanced imaging endpoints, and we do those as well. Vertebral fracture assessment you can do on the DXA. It allows you to look for fractures alongside the DXA assessment. It's not a standard osteoporosis endpoint, but it is available. And then there's more advanced analysis like hip strength analysis, finite element analysis, quantitative CT, high resolution peripheral quantitative CT, HRPQ-CT, looking at bone strength, cortical structure, and fracture risk. So recently, the FDA qualified surrogate endpoints for postmenopausal osteoporosis as a percent change from baseline of 24 months using hip DXA. So while that is now the new, what the FDA accepts, they no longer require a fracture trial. However, fracture outcomes remain important for comparator strategy and payer expectations. So if your drug, if you're trying to compare, or if you're doing a biosimilar, you're trying to compare, if you have a new osteoporosis drug that you need to compare to a previous drug like denosumab, it might make sense to continue doing the fracture outcomes so that you have a comparator, and the agencies, the different payers will pay in the various countries. So so DXA, just to wrap it up here, DXA is used in osteoporosis and bone health, obesity, the GLP-1s, and oncology and bone safety. So DXA is it's very subjective analysis. So it's vendor inconsistencies. So there's two types of DXA machines. There's lunar and hologic. They have different physics, different calibrations, and software references. And so how we control that is a vendor-neutral pipeline, cross-calibration per study, and harmonized reporting. So what DYNAMIKA does is that the DXA images are not DICOM, they're proprietary images. So you can only see the DXA images in the proprietary software. However, you can upload, just as a DICOM image of a CT, a PET, into DYNAMIKA. So the site uploads the DXA image in DYNAMIKA. There's a validated push into the DXA software, and then there's a validated upload of data. So everything stays within our system. There's no moving data around. It's all validated 21 CFR Part 11 compliant, and tested through many studies and audits. When you're looking at body segmentation, you want to make sure that the ROI, the placement of the segmentation is consistent from baseline. So we have seen in instances where sponsors have allowed sites to do in a GLP-1 obesity trial local assessments, and could not see any difference between placebo and drug. Then they gave it to us. They gave us the raw data to resegment everything, and in the end, they did see difference and can continue with the trial. So we feel it's important to maintain that consistency to use a provider like IAG. And then obviously we're constantly looking for calibration or drift in throughout the trial. So we ask the site to upload their instrument quality control data, and we see over time if there are issues or drift in their scanner performance, and we can provide that detail to the site and help them manage that situation. We also have processes for if a machine changes. Obviously, we don't want that. That could have an effect on the patient data for that site, but we do have process to manage that, to keep the consistency and provide the data outputs for the trial. So going back further, how do we do that? So we provide the site with a questionnaire, and we identify which scanner they're using and what version. So IAG keeps the most recent version always validated at IAG. We keep the validated version. Therefore, any lower version can be seen. So if a new version comes out and the site has it before us, we won't be able to see it. So we maintain relationships with the DXA vendors, and when a new version comes out, we do the full validation and test of that version. So there's no issues with data being uploaded that we can't see. We asked them for an instrument quality control baseline, so we have something to go off for the life of the study. The sites upload the data, no issues for them. Once it comes in, DYNAMIKA knows whether it's GE or Hologic, so it automatically pushes the data to the right instrument. There's no manual intervention. There's no manual pushing data, so the DYNAMIKA knows where it should go, and it goes. The team are notified that the DXA image is ready, and they go in and do the QC, and then they do the analysis. So in the QC, they're going to look for positioning. They're going to align the ROI and the segmentation based on years of training and documentation and SOPs at IAG. So everything is standard. All of the DXA analysts do it the same way through training and documentation. And then we can print reports that not only include the BMD. So in an osteoporosis trial, you might have T-score and GENANT combined eligibility. So IAG can do both of those, BMD, T-score, and GENANT within 3 days, less than 3 days. So our average is 1 to 3 days, 97% or less than 3 days. And this is based on a large phase 3 trial that we just completed, large phase 3 osteoporosis trial that was just completed with 1,200 eligibility scans, and the drug has been approved in the EMA in Japan. So for obesity and GLP-1, so why is DXA, why does it matter? Why do sponsors want to use DXA? Why do the regulatory agencies expect it? So in a whole body composition DXA, you can see lean mass, so you can see bone mineral density, and then so it's an easy way in a 24-week trial to see if the patient or the subject is preserving their muscle and losing body fat or VAT. So in the whole body DXA, we're going to look at things like fat mass, lean mass, VAT, and android to gynoid ratio. We'll look at the BMD to see if there's an effect on the bone. So we're going to look at the total hip, femoral neck, and lumbar spine. And then sometimes we can go into the more less than standard imaging like TBS, HRPQC, and QCT. Those would be in an obesity trial. Those would be probably exploratory, and we could discuss with you how that could help your study, look at a mechanism of action, or sometimes in the obesity trials, we'll go further and do the MRI and look at thigh MRI or liver PDFF, looking at muscle and fat in those locations. And now I'd like to transition to my colleague Romiesa to talk about X-ray.
Thank you. So I will be speaking a little bit about X-ray, of course, the most well-known and well-understood imaging modality in musculoskeletal studies. So on the left side of the screen, you will see just a small subset of the validated scoring systems supported in DYNAMIKA for X-ray assessments. We use a range of established methodologies depending on the indication. So if we're looking at osteoarthritis-related studies, of course, we have KNL grading, which is required for eligibility requirements. If you're starting then to look at these patients over time and see drug progression and any safety markers, you'll start to look at joint space width measurements, so if there's any narrowing within the joint space. And then if we're looking at osteogenesis imperfecta or osteoporosis-related studies, using the GENANT scoring criteria, one for understanding any symptomatic or asymptomatic fractures that they may have at baseline to give that categorization of that patient, and also using it as a safety trigger and endpoint trigger for understanding if there are any new or worsening fractures throughout the course of the study. So together, these scoring systems give us both a qualitative and quantitative way to kind of track your disease progression. Now, on the right side, we want to emphasize something that's just as important as it is to choose the methodology within your study. The data is only, or the data output is only as good as the images that you acquire and the quality of your data. And that's why it's super important to ensure that we're able to acquire high quality data throughout the course of the study And we do this through several key practices. Of course, when you're looking at osteoarthritis, having positioning devices at the start ensures that you're able to see the flat medial tibial plateau, and you're able then to track that across visits. You're able to ensure accuracy, reproducibility of that exact image over time, both within that patient and for all patients within the study. Of course, patient positioning, preparation, making sure that there are clear boundaries for upper and lower markers. If you're looking at the spine, making sure that the projection is the same, the images are required in the same manner. That is locked in, and it goes without saying, the same laterality, of course, over all visits. All of these things are supported through the training and qualification process and through the real-time QC that Olga mentioned that we perform as well. And finally, central reader quality control. As much as it's good to have the data outputs and the reading methodologies, it's also super important to make sure that the eligibility and endpoint accuracy is also maintained throughout the study and how that is done throughout the course. I will then now hand it over to Olga, who'll speak a little bit more about the MRI aspects.
I'm on mute, so I'll unmute myself. Thank you, Romiesa, very much. Thank you, Michael. Just before I talk about MRI, which is an important modality, let me answer a question which Fatima brought, which is extremely interesting question just here on the chat. And this is about using AI in clinical trials and how FDA would look into the findings generated by AI. So Romiesa spoke about joint space narrowing, which is a very interesting, very time-consuming measure, which is required in many osteoarthritis trials to support claim of disease modification. To conduct that measure, a reader needs to place 2 lines around the knee joint and then measure space. So of course, AI would do a much better job, especially given that X-ray of the knee is so well studied. However, you can't just purely submit the AI-generated measures as part of your phase 2 or phase 3 submission. So we integrated a pipeline into DYNAMIKA that allows us to pre-process images using AI, and then a qualified reader with the right accreditations would sign off on the report. We also allowing the reader to modify the AI-generated results. I think overall that gives us ability to use AI to keep the cost down, to increase efficiencies really, especially in those highly time-consuming measurements. But at the same time, you still have a signature of the reader, which is a recognized expert in the industry. That's one of the ways of integrating AI into clinical trials. MRI actually brings even more quantification and various functionalities to really look at things beyond just damage, which is visible on X-ray. We have several tools, we have several methodologies, worms, blocks, smokes. Those are radiologically driven. So you have a form in DYNAMIKA, a reader is interacting with an image, and it's quite a lengthy form where each kind of sub-region of the knee joint is divided into smaller regions, and then we're looking for changes in cartilage, bone marrow lesion, meniscus, any kind of changes, and that's manually scored from 0 to 3, and then the overall result is a total score. So there are attempts to make this more automated or more pleasant to the reader to do, because it takes about an hour for each of the scoring. It's attempts, so not yet validated. So we're still deploying a very reader-specific, reader-driven scores in those trials. Now, moving a little bit to quantification, more interesting imaging modalities, imaging sequences within MRI. Those are related to cartilage thickness and volume, cartilage quality, and contrast of the synovitis. So changes in contrast of bone, changes in synovitis of bone marrow lesions through dynamic contrast enhanced MRI. So these are processed generally with automation. When you're looking at synovitis and changes in synovitis, this is for inflammatory changes within a knee joint, it's nearly impossible to place a region of interest that will be accurate enough to depict the volume of synovitis. So we deploy proprietary methodologies which are again, part of our library on DYNAMIKA to map synovitis and give it a much stronger presence in quantification. So we can spot early changes, we can quantify smaller cohorts and actually see statistically significant changes. As a company, we are fortunate to have worked on pretty much every anti-inflammatory drug in RA, PSA, starting with Humira, so it's 2007, as early as that. And those methodologies stayed with us, and we're deploying them across multiple trials. So moving from inflammation into cartilage and cartilage quality. So as we just literally coming from one of the largest osteoarthritis conferences happened last week, WARSI, these measurements are becoming much more important. And sometimes they're not just important as efficacy, early efficacy measures, they're also very important in patient stratification. So there is a huge push, and I think maybe I move to the next slide, Romiesa. So there is a huge push to really start thinking about mechanism of action and develop drugs which are tissue modifying, not just disease modifying. So I really enjoyed presentations last week, which really speak about changes in paradigm from just purely looking at a single joint space width or changes in the cartilage to really understand your mechanism of action first and foremost, to really phenotype your patients before you start recruiting them and really understand from earlier data what would be most appropriate. And then using that information, build an imaging protocol that will support patient recruitment, patient stratification. That imaging protocol will be then implemented at the sites where we're locking parameters, depending on the scanner type. And once you have opportunity to acquire images, then you can choose the scoring systems which would be most appropriate. Of course, all of this needs to be thought through as you design the trial, because the endpoints can only be extracted from a certain image type. So if you have forgotten to have contrast, gadolinium contrast, you probably going to fail to measure synovitis or bone marrow lesion accurately. You will have some more surrogate measures like diffusion synovitis. So that needs to be thought through. To use AI as a segmentation for a pre-read, that's becoming a must-have tool. So we often deploy automatic segmentation of tissues, of parts of the hand, knee, shoulder, any other modality, just to have that information available to a radiologist to conduct the scoring systems. And we move to the next slide. Thank you very much.
So just a couple of case studies that we want to mention. I mentioned previously, we just completed a phase 3 postmenopausal osteoporosis trial, looking at DXA and spine X-ray. The primary endpoint obviously was DXA and spine X-ray, and it was submitted to the regulators and approved in Japan and Europe. And then a phase 3 obesity trial looking for fat loss in the BMD and composition, and also MRI of the thigh muscle. And Olga, you could mention the case too?
Yes, happy to. So obviously there is a number of recommendations to go beyond joint space widths. This is really towards building disease modifying drugs in osteoarthritis. So we are looking at targeting different mechanism of action, and the endpoints should be appropriate. So there is a variety of studies starting now, and I like the new vision of osteoarthritis drug developers to really integrate better screening, more stratified populations, and look at the endpoints beyond just simple X-ray and joint space widths. And last case study, Michael, to you.
So IAG can support your end-to-end imaging trials by helping you identify the imaging endpoints necessary to get the answers that you need. In terms of efficacy and safety, we work closely with the FDA and the EMA. We have a team of people that do that. Our KOLs are very experienced with those agencies. And during early considerations of what the imaging strategy is going to be. IAG will qualify the sites and train them to have consistency across all the images and the visits. We developed the imaging protocol that will also allow for consistency across visits, even if there are different instruments at the site, different MRI scanners, different DXA scanners. We provide data management and QC, edit checks within DYNAMIKA, and blinded central review with our expert readers.
Super. So we have a few questions on DXA, Michael. Maybe it's worth you looking at the chat. And Shreya, why don't you pick up on DYNAMIKA? So we will have a look at the few slides related to DYNAMIKA and then live demo. So we have some time, and then we will address the questions.
Sure, sounds good. Yeah. So just before we jump into the demo, I think it'll be good to give a little note here about compliance because it's pretty important in clinical trials, right? And we have a few frameworks here already set in place for studies that run with us, as well as how the data flows in. So we have 21 CFR Part 11 compliant modules. We are SOC 2 Type 2 compliant, and we have these ISO, GDPR, HIPAA related frameworks in place. So right from image acquisition, we have our validation checks. We have dedicated storages for studies in place. When our readers come into the platform, we ensure that our ECRFs, or the case reporting forms, have validations in place to ensure that they are guided in the reading that they perform. And finally, we have our dashboards and our dedicated data management team who helps us basically wrap up data packages for submissions. If you move on to the next slide, that's basically a little introduction to the demo. And I will now walk you through just a basic workflow that you're seeing on screen, right from trial setup, how we upload images, how a QC is done, how we read, and the dashboards at the end. Okay. So let me take over the screen. I'll share my screen and let me know once it's visible.
Yeah, we see it, Shreya.
Perfect. All right, thank you. So you're seeing the login page for DYNAMIKA. And I'm now going to log in as one of the roles, that is a project manager role in DYNAMIKA. And this role basically helps to configure the studies in our trial, right? So depending on the protocol, what you're seeing here is a list of clinical trials. So not only would you be given an invitation to come onto DYNAMIKA, you need to be part of the clinical trial to see data for that trial. And so we are seeing a list of clinical trials. And when we have to configure a particular trial, we need to give in certain information about it, like the name, the sponsor details. We also provide an option to define which storage location you want to save your data in. So if it's regionally specific, we can provide that here. And some assumptions about what's the start and dates and when are we expecting subjects. So all of this really helps to build a dashboard that I'll show you in a few minutes. In the configuration, we also have very important our imaging labels, which tell us what kind of modalities we're going to expect. And these labels are going to be used by the imaging specialists in the workflow to label the images. Right. We have the visit schedule here, so you can define different cohorts, different visits in every cohort. You can also define subject ID patterns for these cohorts. So it's super configurable when you want to define the visit schedules in the study. We also can define our data transmittal forms. So these are forms filled in by the site user when they start uploading data onto DYNAMIKA. So even that is configurable with every visit in here. And this is a reading config. So this is actually like quite an important engine which defines how the data is going to flow. So right from what labels we need to generate our reading tasks, if it's a single read, if it's a 2+1 adjudication read, all of that gets defined here as separate tasks. So this kind of defines the order of how the workflow is going to go in the study. And obviously, we need to define certain sites who are going to participate in the study and who are the team members of the clinical trial. So as you can see, there are these different roles here. So all of these members are part of the clinical trial, and we can invite them here. And we also have our dashboard. So the project manager as well as the sponsor sees the same dashboard, and these are dynamic. So as data is coming into the system, you will see the changes on the dashboard, and you can drill down across different subjects and see at what point of the workflow they are in, right from upload, QC, reading, right? So we can have a look at an overview of that. If a study has some eligibility reads, we can have a look at eligibility reads with every sites. And some other dashboards regarding uploads with every visits, how many were QC'd, how many were read. So all of that metrics is available for our sponsors to look at. Available, yeah. Yeah, we talked about compliance, and so we also have audit trails in place. So you can also see timestamp events of the team members who have performed any action in the study. So you can have a look at the audit trails within DYNAMIKA itself. And I think I'll move on next. I'll quickly log back in as a site user. This is the next role in the workflow. So the site user would essentially start uploading data into the platform. So let me just put in the credentials here. Okay, so I need to select a clinical trial I'm part of, and what the process for this is basically you need to first create a subject to start uploading. So the site user would select the cohort, would give in a particular subject ID and the month and year for the subject, right? So all of this is coming in from the configuration I showed in the previous step. So once a subject is added, we begin the upload process. It's a stepwise process. So the first step is selecting the time point for which they would be uploading the images. Like we said, we allow different types of file formats, and this particular study is allowing DICOM and DXA file formats. So if I select a DICOM, I'll move on next. And this is the EDTF form. So they need to fill in, again, the month and year of the subject, when was the images acquired, and who is uploading the images in the platform. And what the site user would essentially have to do is zip up the DICOM files and drag and drop them in the model here. And they upload it. And that's it. So now as I'm speaking, what's happening in the background is it's kind of verifying if it's a correct file format. There was a question on the chat as well about pseudo-anonymization. So that is also happening in the background, where all your patient-sensitive information is being stripped out. And all of that is automated and happening as I speak in the file processing step here. Right, so I'll move on next. This was the site user, how they interact with DYNAMIKA. And the next user in the workflow is our imaging specialist. So I log in back as an imaging specialist role. Okay, so as an imaging specialist, what they see in their workflow is they'll see the list of labels that they would ideally assign to the DICOM, and they can then generate the reads in their workflow. We spoke a lot about how we have integrated DYNAMIKA with the DXA workstations. And so essentially the imaging specialist would now send in the DXA files from here, from this particular model. The analysis will be done on the DXA workstation, and we can pull it back into DYNAMIKA in the same view. So we kind of sync the results back from the workstation here. And so what you're seeing here is essentially the results that is coming back from the workstations, where you can see the BMD, the composition scores from the reference population there. And the imaging specialist would assign labels here. So this is going to determine what goes to the reader for a final review and what should be included in the final report then. So yeah, that's basically the little workflow, how they send DXA scans and receive back results. We have genome scores as well. So for that, they'll have to open up the X-rays. And this is our image viewer within DYNAMIKA. So essentially, they can see the images, the DICOM images that were uploaded, and they would assign the labels here in the same viewer. So let me move on next and finally show you the reader workflow, and we'll get an idea how the reader tasks look like for them. Okay, so yeah, so as a reader task, this is basically what they see. They'll see a list of reading criteria that needs to be performed. So we spoke about DXA eligibility, DXA scoring, GENAN forms here. And maybe I'll open a DXA eligibility form, and you can see the different regions that were labeled in the previous step by imaging specialist, along with the BMD and the T scores coming here. And so the reader would give like a final sign off if they want to include these results in the final report. You can also see the machine manufacturer it's coming from the Luna workstation, and some other general information about the subject. And so once they sign and submit, we can have a signed off copy of this reading. So this was the DXA eligibility. I'll also show maybe the GENAN scoring form as well. So that will take us back to the viewer, and they can see the X-ray images here, the GENAN scoring form, which is your vertebral fracture scoring form. They need to grade it from 0 to 3. And once that is done, they would sign and submit it again by entering their DYNAMIKA credentials. So this is the signing off with the 21 CFR Part 11 compliance. And once that is done, once the reader submits the form, they can still view the readings in their end. The only difference being they can't modify anything because it's already submitted. And so they can only view what was graded and then see it in a view only mode here. Yeah, so that's basically it. That's the reader workflow. And finally, at the end of it, you would have a ready report in DYNAMIKA, which would look something like this, that captures all the analysis that was done by the reader, signed off copy, along with all the scores that was captured. Yeah.
Thank you, Shreya, very much.
Yeah.
You make it very easy, but now if you can imagine that these workflows are scaled to hundreds of sites, multiple readers, multiple project managers who are simultaneously accessing cloud-based platform and going through their workflows and doing their jobs, that becomes a real factory. And few things, which we're particularly proud of. So for the sites, it's a really less burden. So the sites already participating in their spare time, so to say, and for them having a simple workflow where nothing needs to be done apart from drop of the images, acquisition of correct images, of course, that really works. If the site working with us on multiple studies, which of course happens, so you saw Shreya was logging in as a site, and there was 3 different options. So the site has a unified login. So they just have the same platform and they select the right trial where they will be uploading images. The same for the readers. So if reader works on multiple studies, and we have some big KOL readers which work on more than few studies, the same thing. So we make their life as easy as possible. And lastly, of course, for the sponsors, that's very important. All of the studies are running in a single platform. So we do not clone DYNAMIKA for every study. It's not a clone of a software. It's a single multi-tenant software where each of your study is visible, and as a program manager or as a project manager, you have an opportunity to really manage the entirety of the pipeline. So you have full visibility. So you could say, okay, the site is doing a good job on this study and not such a good job on this study, and so on and so on. So I have to allow 10 minutes for answering some questions. Thank you very much for throwing some interesting questions. Michael, Romiesa, do you want to pick it up?
Can I
Yep.
answer some of the DEXA questions?
Of course, please.
Yeah. So one of the questions is, for participation in a clinical trial, what technologist certification is required? Is it manufacturer, core lab, ICSD, or other? So if you're referring to the site, so we don't require any type of certification. We just ask if they're experienced, and we train them to scan the images in a standard way. Our technologists, our readers are trained by the manufacturers. So I don't think for the site level, we don't require any type of certification. For obese subjects, is BMD value compromised by mass of soft tissue? So yes, sometimes we will see change in BMD as the weight fluctuates up or down based on, but for us, it's not a matter, we only see it at a time point. So at the statistical level, where you might see change at the level of the study, that would happen at the end of the trial. So we're not looking from day to day or visit to visit what change is actually happening. We're just reporting the change and comparing it to previous study. I will say that there are, in the obesity trials, you may have instances where the patient is too large to fit on the scanner. And we do have a process for how to manage that and how to scan the patient in a standard way across visits so that we can assess the data. Is there now a validated or scientifically accepted phantom for the use of body composition measurements? So yes, so what we ask for when we're doing body composition, we ask if the site has one of two body composition phantoms. And if they don't, we ask them to acquire it. So Hologic has one and BioClinica has a body composition phantom. So we ascertain upfront if they have that phantom. And I'll answer one more, I suppose. Must DXA scans be pseudo-anonymized prior to uploading to DYNAMIKA, or is it personally identifiable information automatically stripped during the upload process? So in DYNAMIKA, so DXA is proprietary software. So if it's DICOM, we automatically strip the PHI from the DICOM header. In dynamic, the DXA will come in. We train the site how to label the DXA image so that it does not contain PHI. However, we do check it for PHI when it comes in for QC, and we do remove that at the QC process. We also will send a query notifying the site that there was PHI, and in the future to follow the process of anonymizing the DXA scan before it comes into DYNAMIKA. I guess one more. Can I do one more? I'll do one more. Can DXA analysis be performed directly within DYNAMIKA, or must it be completed in a vendor-specific platform? So it is completed in the vendor-specific platform. However, it's an end-to-end solution. So the site uploads the DXA to DYNAMIKA, no different from their MRI, their X-ray, their PET scan, their CT, whatever else is in the trial. It automatically will send the image to the correct platform system. So it will go to Hologic or Lunar. The analysis is done, the results are automatically sent back to the database. So it is done in the vendor-specific platforms, but for the site, there's no difference. You will upload in DYNAMIKA as with any other image. I think that's all the time we have for questions. I can answer other questions via email or later, but
maybe
I'll leave
Romiesa,
it
you
there.
can pick up one of the questions. So how do we manage adjudication amongst readers? I think that's very interesting question.
Yeah. So this is very heavily depending on the methodology that you use on your study. So we've discussed a lot of the methodologies here, and also with regards to the endpoint. Is an eligibility criteria, primary, secondary, exploratory, and also what phase of the study that you're in. So a lot of the methodologies that we spoke about today are implemented within our platform and can be done in multiple ways, both a single read, double read with adjudication. In terms of monitoring adjudication amongst our readers, there are very well-defined discrepancies you could see. So with KNL, it's a very subjective scoring criteria, and therefore the percentage of adjudication can be quite high. And therefore we have monitoring processes for that. But there are other criteria where adjudication may be triggered depending on the presence or non-presence of a fracture, and depending on areas of interest. So those are all aspects that are very well defined within our imaging charter. We will also have quality monitoring plans for all the studies where we see trending patterns throughout the course of the study. So we would make sure that we review and visualize the adjudication rate amongst our readers to make sure that they're reading as expected.
Perfect. Thank you very much. Well, it was great to have everybody online. Thank you very much for being with us, and we hope you will send us more questions by email. You have our email address just at the bottom of the slide, as well as the website. Obviously, demos of DYNAMIKA more specific to your indications and your studies are available, so our team would be delighted to engage. Thank you very much, everyone, and we hope to see you at the end of May to talk about new topics and new subjects. So there will be another webinar. Thank you.
Thank you.
Thank you. Bye.
Thank you. Thank you very much. Thank you.